All right. Here we are for lesson 8, group problems. Nathan and Joshua and Jacob are over there. You should know us by now. So, question 1. We've got these two chromosomes. One has an inversion from the V to the R allele. They twisted. And so we're trying to figure out what would happen if there was a translocation between the S and, if there was a crossover between the S and T with these inversions. So the easiest way I have to figure that out is to write them both out. And then since the S and the T are inverted, write the S and the T the same way as the first allele so that they line up, that the S and T line up. And then once you have that, it's pretty simple from there because all you have to do is your 4 gametes are going to be the first one normally and the second one normally. And then when you do the crossover, you just start at the left until you hit that line, and then you jump up. Or if you're going from this one and then you hit the line, you jump down. And so number 3 is dicentric because it has 2 centromeres in it. And then 4 is acentric because it doesn't have any. And so again, just for 3 and 4, what I did for 3, you start at the top one, work your way left, and then once you hit the crossover between the S and T, you jump down to the T on the bottom line and go there. And then for number 4, start at the X, go to the S, and then jump up, and then the T to the X. And that is how you do number 1. And then number 2 is fill in the blanks. So non-disjunction in meiosis 2 produces 50% aneuploid gametes, whereas non-disjunction in mitosis can produce a mosaic phenotype. And then someone who has suffered a paracentric inversion produces a dicentric chromosome, acentric chromosome. So kind of going back to this one, this one's dicentric because we've got 1 centromere here and 1 centromere here. And so the paracentric inversion is left out of that. So you end up with the dicentric, or I should say the inversion from the, the R to the V does not involve the centromere. So that's a paracentric inversion. And as you can see by the problem, it produces a dicentric and acentric chromosome. And then based on the reading, we chose Turner syndrome as an example of monosomy, while familial Down syndrome is an example of a disease caused by a Robertsonian translocation. Human liver cells can be 4N, 8N, or 16N. And then terminal deletion on chromosome 5 causes Creux-Duchat syndrome, while an inversion on the X chromosome causes type A hemophilia. And a duplication is a change in chromosome structure that can be potentially beneficial to an organism or an entire species. And why that is because, for example, the globin family, that gene has been duplicated several times. So mutations are able to occur in that gene without being completely detrimental. And so we have more genetic variety, which is always beneficial for a species. And if we have that duplication, if something does get messed up and there is some nasty mutation that comes out of that, or it makes a nonsense protein, then no harm, no foul. And then for the last one, when a translocation is formed, the alternate segregation pattern can produce viable gametes and offspring. And that's it for me. On to Joshua for number 3.
Thank you, Nathan. Now I'm going to teach you number 3 and number 4. And number 3 was pretty simple, so you just have to For number A, we have differences in chromosome number and structure. So for the Turner syndrome, that means you have a monosomic individual. So he only has one X, she has only one X chromosome and 45 chromosomes. Jacob syndrome, you have two Ys, so that would be 47 chromosomes. Cri-du-Chat syndrome is where chromosome 5 has a terminal deletion on the P arm. So you would have 46 with a missing part there. And then familial Down syndrome, you'd have a Robertsonian translocation. So you'd have 46 chromosomes. And you still have chromosome 21, but one would fuse with a 14 chromosome. So that would still be 46, not 47. And then type A hemophilia is an inversion of the X chromosome, and so that would be 46. And then which of the following terms is not associated with Marie's tooth disease, type 1A? And they all are not, except duplication is. That's how you actually get that disease. Which of the following terms is associated with Klinefelter syndrome, aneuploid, and trisomy, and non-disjunction, how you get trisomy. So those three would be associated with. The rest are not necessary. And then which of the following terms are associated with type A hemophilia, and none of them are. However, inversion is associated with it. And number 4, this is a rank of a question. So we have one pair of homologous chromosomes in a mouse, and then we have a reciprocal translocation. And the picture entry here is like this. So they basically cross and switch those genes, MNO and DEF. And then identify the allele composition of the gametes produced by alternate segregation. So the way you do these, the alternate actually produces viable offspring. So when you do the cross and you go diagonal, and then you come over, you would have half the gametes would be normal and half the gametes would be unbalanced, balanced translocation. And when you have that, you have 100% normal individuals. And then, when you do the other two, so C and D, you would have adjacent 1 and adjacent 2. And those would produce not viable offspring. And so you can kind of see how I did it there. And so you'd have the DEFs and the MNOs, and then you would switch those. And then here's kind of the alternate pathway. You would have 2 normal, 2 translocations, and then you'd these guys are being silly over here. And then adjacent ones, you would have normal with translocations, and adjacent 2 would have normal with translocations. So those would not work. You would not have the necessary alleles to survive that, so and be born.
Indubitably.
Yes, and this is such a rancor right here. So, so if you want the full questions there, you can kind of defer that from the notes there. All right, now I'm going to turn the time over to Jacob.
So I guess I'm going to be doing the last problem, which is the cat problem. Just move it over here so you can see a little bit of the work that's being done. It was pretty easy. So the first one is going to be asking, cats have 38 chromosomes. How many total chromosomes would be found? The following types of cat. So the first one is a triploid cat. So triploid is where you have specifically 3 like sets of the gene. And so we know that they're diploid to begin with. So we take 38 divided by 2, we get 19, and then we multiply 19 by 3 and we get 57. So it would have 57 chromosomes. A cat that is monosomic for chromosome 12 would be 38 minus 1 equaling 37 because it should have 2 chromosomes in each one. Monosomy is where you only have 1, so you have the absence of 1 chromosome. A cat that has suffered a terminal deletion on chromosome 7 would have 38. The terminal deletion is only usually a deletion on on a portion of the cell, and that will bring adiabatic changes to the cat. But for just the number of genes, just the number of chromosomes will be exactly the same, with that 7 portion being missing a little bit. A cat that has suffered Robertsonian translocation between chromosome 17 and 18, it will be 37 because Robertsonian translocation is where 2 chromosomes will fuse into 1, and so you'll take 2 and you only have 1 left, so that's 1 minus. And then finally, a cat that has suffered a reciprocal translocation between chromosome 6 and 9, and that will also be 38 chromosomes. There will be no change with that. However, the 2 chromosomes will swap some of the material, and so that will be a little bit different. Thank you very much for listening, and have a great day.
That's really good.
Oh wait, no, wrong one. I clicked stop video.